Hi, everyone. Thank you so much for joining us. My name is Amanda Abraham. I'm the Director of Development here at the Peconic Land Trust, and I've been assisting with the fundraising efforts for this project. So just a couple of ground rules before I hand it over to John. Just please keep yourself on mute and use the chat function with any questions. I am going to be monitoring it the whole time. Once the presenters are done with their section, we'll open it up for 5 minutes of questions in which I'll ask the presenter the questions that you put into the chat box. If we don't get to all of your questions, I'll be sure to write them down, and we will definitely connect with you after this event to answer anything that's outstanding by the end of the week. And so beyond that, I'm going to just hand it over to John. Thank you for joining us.
So welcome, everyone. Hope you had a good summer. Today being the swan song of summer of 21. And, I think we have some interesting news to share with you tonight and some results, preliminary results from some of the research that Ron and Molly from Cornell Cooperative Extension have generated these several months. And also we'll get an update on the water quality of the pond from Chris Gobler, as well as next steps as we look to the future. So I'm not going to belabor my time here, but let's get right to the meat of the evening. We're going to try to get this all done within an hour. If we go over a little bit, that's fine. Hence Amanda's help in facilitating the questions. We're going to have the questions about 5 minutes or so after each of the presentations. So we'll have 2 presentations tonight. And I want to kick it off by introducing Ron Paulson and Molly Graffam from Cornell Cooperative Extension. And I'm not going to say anything more. I'm going to let you introduce what you've been doing, and many of you on this Zoom know from our previous session, but you may want to give a little bit of an overview, and I'm sure you will.
Sure. Well, thank you, John, and all the support from the community and the opportunity to go out and do this investigation of the pond. It was a big embayment-wide study that probably one of the largest we've ever undertaken, and some challenging at times, but I think what we learned and what we're going to share is very insightful, and I think it will help focus where we want to go next. So I'm going to pass off to Molly. She's going to kick us off, and then I'll jump in later in the presentation. Thank you, Joe.
So I just wanted to share my screen, but Josh, do you think you could give me the ability to screen share?
Just made you co-host as well.
Okay, that worked. Josh, can you see our slides?
Yes.
Yep.
Great. So I'm going to kick us off with sharing some of our preliminary results from our project on identifying and reducing groundwater nitrogen input into Sagg Pond. I'll start off with some background information, and then we'll go over some of the methods, the preliminary results, and then some of our next steps. So for those of you who are familiar with the subwatershed plan, you may have seen this before. What I'm showing here is the model output of the Sagg Pond subwatershed region. And what you can see is that the groundwater travel time for the groundwater contributing area to the pond ranges from 0 to 25 years. So the different colors here on the map represent the different groundwater travel times. And This means that nitrogen, the nitrogen concentration that we measure at one time point is a value that reflects recent nitrogen inputs as well as legacy nitrogen inputs during the groundwater travel. And nitrogen that was added to the groundwater 10 to 25 years ago, such as in the yellow areas, is actually just now entering the pond. Or to put it another way, since the groundwater travel time is relatively slow, nitrogen added to the groundwater today in those areas will continue to enter the pond for decades to come. And this makes land use very important, and land use in the area, as you all know, is primarily residential as well as agricultural and some commercial. As you're likely aware, Long Island sandy soils are very susceptible to leaching nutrients like nitrogen and other pollutants, making the underlying aquifer susceptible to contamination. And this makes land use and the chemical makeup of groundwater within the subwatershed critical for improving and maintaining water quality. And groundwater is contaminated with nitrogen from several sources, including septic, fertilizers, and atmospheric contributions. Those are concepts that you might be familiar with, but a new concept that we'll cover tonight that you may not be familiar with is the mechanism of how groundwater is impacting surface water bodies. And groundwater enters Sagg Pond and other coastal water bodies through a process called submarine groundwater discharge. So what you're seeing on this map is also a model output, which we looked at and can use to help focus our groundwater survey work. So the map is water table contours, and what we can see is that the groundwater travel is actually perpendicular to these blue lines, which are the contours. And moving closer to the shoreline, the blue contours get even more parallel to the shoreline. So the flow is really perpendicular and going into the pond. And what's interesting is that the distance between these blue contour lines tells you about the modeled speed of groundwater flow. So the closer the lines are together, the faster the rate of groundwater flow. And we can use this to start to focus our groundwater survey work. So just to give a quick recap of some of the important terms and the key concepts here, there's a very simplified schematic on the left showing like a side view of some of these processes. And we have the water table height, which is higher inland compared to near the shoreline. And this change in water table height is really forming a pressure gradient. That is what naturally causes the groundwater to flow towards the water body. And this process of the water moving into the water body is called SGD, submarine groundwater discharge. And what's interesting is that at Sagg Pond, that water body height can also change. So that can influence the pressure gradient and also the speed of groundwater flow. And just so we have some terms here that you will understand as we're talking, when we talk about sediment, we're really talking about the bottom of the pond, and the pore water is the water that's within the sediment. And yeah, I think those are most of the key terms. And then the key concept here that we'll try and explore and show with our data is that the pore water with elevated nutrient concentrations discharging from the sediment directly into the surface water is going to impact surface water nutrient concentrations and surface water quality, especially in areas with significant groundwater discharge. And groundwater discharge has been identified as a major source of nitrogen to surface waters. And of course, the nitrogen pollution is then directly related to water quality issues. So sometimes it is kind of hard to understand what this looks like in reality. So I just wanted to show a quick video clip here. Hopefully you can see the water percolating up through the sediment in the center of the slide there. So this video was taken at a different site, but this process is also happening at Sagg Pond. And you can see this process happening, if you walk along the beach in some areas at low tide. you'll see little riverets forming in the sand and water flowing towards the ocean. But in other areas, the seepage is kind of coming up from the bottom sediment, and this is happening, but in some cases you can't see it because there is water already overlying the sediment, so you can't really see that process. But in this case, we were able to see it and take a quick video. So we're going to move on to some of the methods that we use during this survey. So here on the left, we have a map of Sagg and kind of a zoomed out map so you can see some of the watershed and the land use areas. And most of our data today will actually look like this map where we have dots on the map to show the stations. And when we get to the results section, we'll also have different colors that represent the chemistry of the pore water. But here I'm just showing a red dot for every station and a yellow dotted line for a transect. So we began this survey earlier in the year in April. So far we've accomplished 20 transects, and I think that's pretty much where we're gonna stop. We've covered most of the pond now, and we're really getting into some of the data analysis now. But as you can see on the map, there's the red dots that are kind of scattered around the whole shoreline of the pond, as well as within the transects. And we've covered a total of 184 pore water stations and collected 122 pore water samples out of those 184 stations. So quite a bit of sampling and quite a bit of work done this summer.
Yeah, so
Thank you.
Yeah, thanks, Molly. So I'm going to take the next few slides about some of the methods we use to collect that data of the pore water and the rates of discharge within the pore water using seepage meters and such. So on the left is a pore water device we use. I'll talk a little bit more about that in the next slide, but basically at every one of those stations that you saw that red dot previously, we collected a variety of parameters to help us to understand where this submarine groundwater discharge is offshore and along the shoreline. And those parameters included conductivity, which is related to the amount of ions in the water, but it's really sometimes easier to understand it as salinity. So you'll see some numbers there that might seem a little strange, but really what we're looking at with conductivity is you could relate easier as salinity. And it ranges really from the salinity of the ocean to really a salinity very, very low that's in your drinking. The other parameters we measure in the pore fluid is oxidation reduction. It tells us a little bit about the conditions in that pore fluid, and those types of measurements determine different species of metals. We also look at pH, which I think most are familiar with, total dissolved solids, amount of ions and such present in the pore fluid, and dissolved oxygen, as well as temperature and other direct conductivity measurements. So the instrument that we use, and nicknamed the Trident, it's something I've worked on for about 20 years with different other collaborators, some with the US Navy, some with Cornell, some with Stony Brook, professors I've worked with there in the past. But basically what you see, that probe on the left, is you'll see like a black plate about 2/3 of the way down. And that plate is the plate that will, when the probe is inserted into the bottom from the boat, that plate will stop it from going any further into the sediment. And below that plate are several probes. And those probes are the instruments that will measure the temperature, the conductance, and collect a sample where we run all these other parameters, including the nitrogen serum. So that's basically the device that we use to evaluate the pore fluids and map out the SGD. We take advantage of this. Why the summer is a good time is because the groundwater is so much cooler than the surface water. So the temperature helps us to map out where these areas are, as well as the conductivity difference between groundwater and surface water. And then, additionally, samples are sent to the lab. The other device that we use, once you understand where the groundwater discharge is, and you have some idea of the values of the nitrogen in that, you can then actually start to go out and measure groundwater discharge rate that's coming through the bottom. And, that's important because if you put those two measurements together, the rate and the concentration, you can start to see areas where that groundwater is contributing a lot of nitrogen or maybe not. So the device on the left goes into the bottom, and on the very bottom, there's a funnel that about a 20-inch funnel that goes into the bottom. And then it goes through these flow tube system and a computer, and we're actually able to measure those groundwater discharge rates in the areas that we determined were important. So I explained a little bit of this already in the previous slide. And so the one thing I wanted to mention was the one thing about flow rates is that they're very, very slow in some areas. It'd be like that funnel is collecting water that it's almost like a drip coming out of your faucet. So, the device is very sensitive. It uses an ultrasonic type of Doppler effect to measure the groundwater discharge, and it's been very effective. And we've used it actually in a lot of different places throughout the country. So I'm happy we can bring it here and add that to the work we're doing here, because I think it's a piece, even though you have modeling and you have groundwater studies, what you really need to understand is the dynamics of that discharge and how that relates to the height of the pond and how that relates to the height of the water table and the various conditions, as well as being able to calculate this groundwater nitrogen load to the surface. So other hydrogeological data that we collected is of the more traditional ways of using groundwater wells. And we did install some wells at the Smith Corner Preserve, which is certainly a site that we're going to consider for potential future remediation. We did put these wells in, we sampled them for nitrogen, and we also used those wells to put different loggers in and measure the height of the water table. And all of that information helps us going forward to have the work we need to design any type of remediation that would relate to treating that groundwater. You have to determine the hydraulic conductivity of the soils, the velocity and direction of the groundwater. So those are more, that type of work we hope to do in the next phase, now that we have areas that we've identified that we know there's significant groundwater discharge with high nitrogen. So I'll start off with some of the results and hand it back to Molly, but we'll go through some of the first cut through the data that we've been putting together. So one thing we did look at that was very informative, besides doing the pore fluid and measurements of that chemistry and other parameters, we also took shallow cores at every station practically. And that was very useful too, because it told us right away, gives you some indication of where the groundwater, might be coming through at higher rates. And typically, when there is groundwater discharge, it's often where the sediments are sandy and gravelly, like you see on the left. And then other areas were almost clay-like. And so those areas are restrictive to the groundwater discharge, or could possibly be deflecting it further offshore. So that was a new type of thing we routinely do now, and we took photos, and we'll have all that data available, in the report. And so here you see the pore water probe. And again, that black plate is where it seals to the bottom, but below it to the left is where the probe was pulling out pore fluid. And it's kind of neat when you have these muckier bottoms that have a lot of silt, that we draw the pore water out of it so efficiently that what happens is it accumulates the sediment and dries it out in a sense, and it sticks to the probe. So it's pretty good evidence that you've collected pore water, even in these very, very fine sediment bottoms. You don't get a lot of volume, but it does a very effective job of collecting that pore fluid. So again, one of the parameters I talked about earlier was this pore water conductivity, which relates related to salinity. But basically the range was 115 microsiemens per centimeter, which is typical of what's coming out of your tap for your drinking water. It could be low all the way up to 2,000, and it would still probably be drinkable. After 2,000, it's no longer really fresh, and you go to more brackish. The highest we saw was about 33,000. Now this is in the pore water, not in the surface water. And that's probably the salinity, well, lower than the ocean, but in the mid-20s salinity, I'd say. So at all these stations, we did all this measurement. And what's interesting right away is if you look at the plot, and the yellow dots are the lower conductivities or fresher water, and the orange and such are mid-level, and then the red are really indicating the pore fluids are much higher in conductance. And what that tells us, there's not a lot of groundwater discharge or groundwater, seeping into those areas. So what we found is that in the northern area, north of the bridge, let's say Bridge Lane, for the most part, it was relatively strong signal of conductance, except in some of the middle areas there. But in the northern areas, very strong freshwater, groundwater drives. And that previous diagram of groundwater contours that Molly had shown, it agrees with that pretty strongly, that would be an area of significant discharge. So moving south along the west shoreline, you could see it was this yellow color, this low conductivity all the way down, almost to where the main body of the pond is. And again, that's in good agreement with what we know through modeling and such. The east side did have significant, areas to the north more, and along that shoreline. But then when you went all the way south to the main body, these peninsulas at the tips of these areas often do not have a lot of drive. And that also is in agreement with the modeling. And therefore, you don't see a lot of groundwater input contributing down there. So right away, we identified areas where the groundwater and the inland sources that are affecting that are contributing. And, you know, we'd want to focus further. So the next slide, I believe, is just some of the results of the nitrate data that we collected at all those stations. And lo and behold, that signal, again, the groundwater ranges, the beige color is like 0 to 1. And then 1 to 5 would be the orange, and then the deeper orange could be all the way up to 13. So what we noticed in this data was, again, the west side and along the shoreline where we saw the fresh signal also had a lot of higher nitrogen values all along that coast. The upper reaches, some of the areas in the upper northern section had relatively high levels. And then on the east side, you know, some of it was lower along that shoreline. So you start putting these pieces of puzzle together, you know, we can really take a look at what the nitrogen, where the nitrogen is mostly entering and impacting, and where we need to focus other work. So so I think I'm going to hand this back to Molly. She's going to take us through some of the seepage data that we collected. But I'll let Molly, you good with that?
Yeah, so now this is showing a map of the groundwater discharge, the groundwater seepage locations where we deployed that meter that Ron had explained earlier. And so these deployments were done after a lot of the Trident work, so that we strategically placed the meter in these locations. And we actually did a little over 50 deployments and found that the seepage rates varied pretty widely. We even found some negative values. So the values range from -75 to even over 200 milliliters per minute. And we found a high degree of spatial variability, kind of even in a smaller area that we studied in several deployments over kind of a smaller area. But we did find that the Trident data and the sediment type also kind of fit into the puzzle and really helped inform the seepage data, and will also help us figure out the area of the discharge zone, which we'll be using in calculations for our report. So in all but one area, the groundwater discharge was positive, meaning that the groundwater was discharging into the pond despite relatively high water levels for most of the survey. However, there was 2 periods during the survey when the ocean inlet opened, and we actually had our equipment out and we were able to measure the groundwater discharge rates, and they increased substantially, and in some cases went from negative to positive in that area where we were measuring negative rates. And generally the discharge rates were higher north of Bridge Lane and also on that western shoreline due to the greater groundwater drive. So our seepage rates definitely matched with the conductivity values that Ron just presented. So I have here a couple of video clips just to give you a sense of what happened when the pond was open. So I'll go ahead and play these clips. So we were there during the dredging, and then the day after the dredging, these videos were taken just in. So as you can see, the water flow to the ocean was pretty strong while we were there taking these video clips. And then what ended up happening is that the water level dropped substantially. So what you can see on the far right is that some of the sediment was exposed, and normally the water level was at like the middle of the frags shown in this picture, but the water level dropped substantially, about like even 2 to 3 feet in some cases right after the opening. And then in August, we know that the inlet closed after about 1 day and the water level increased very quickly, but the water level kind of increasing and the pond filling we do have some data on the water level in the pond, and we are still processing that data, but we do have some information on that, and it will be very interesting to look at how quickly the pond refills. So here I'm showing some data from the June ocean inlet opening. So what you see in the graph is the groundwater flow rate on the y-axis and then time on the x-axis. And I'm showing the location of where this data was collected. That's shown by the yellow star in the map. So this was collected north of Bridge Lane on the western shoreline. And what you can see is that the flow rate was pretty steady at about 50 mils per minute for a good amount of time. And then just before 12 a.m. on June 3rd, this is when the discharge rate rapidly increased to 200 mils per minute, so about 4 times the previous rate. And this coincided right when the ocean inlet opened. So what happened here is that the pressure gradient between the inland groundwater table and the pond water, pressure gradient increased by the pond water level dropping, and that increased the groundwater discharge rate. So again, we were out there in August when this happened, and we had our equipment in a different location. Now, this time in August, we had the seepage meter stationed on the western shoreline again, but south of Bridge Lane, kind of close to the ocean in the opening, actually. And we had data here because this was a really interesting area where we found the negative seepage rates. So this means that the water flow was like surface water was actually going into the sediment. So this was a recharge area, which we were very excited to find, and it was kind of unexpected. But we have some data from just before the inlet opening and then after the inlet opening. And as you can see, the flow direction reversed from negative to positive during and after that ocean inlet opening. And although we were only taking readings at these individual locations, what we can infer is that this type of rapid increase happened pretty much everywhere where there was discharge occurring when the pond was closed. And we know that groundwater discharge is one of the main ways that the pond fills back up. So some of our next steps in this project are to compile and analyze this phase 1 data and identify areas where groundwater discharge rates were high and where pore water nitrogen values were high. And these are areas where significant groundwater nitrogen load to the pond is occurring. And we'll, of course, be preparing a report with some recommendations for phase 2, which is a site characterization for future remediation. So some of the next steps in phase 2 include focusing on these areas where significant groundwater nitrogen input to the pond is occurring. And we've applied for the Town of Southampton CPF funds. That request was made in this past spring. And if the funds are awarded, the results of phase 1 will be used to perform additional, more detailed site characterization in some of the targeted areas that we've found. And this phase 2 will include very focused site characterization work to provide information for the design and implementation of nitrogen mitigation, such as using PRB technologies along the edge of fields. So just to give you a quick introduction on PRBs, they've actually been around for decades and have been used to treat various groundwater contaminants, but they're increasingly being used for nitrate remediation. And they've already been used extensively in the Midwest, US and Canada, to treat agricultural and septic-derived nitrate. And they've recently been installed near impaired water bodies in Suffolk County, along Shinnecock Bay, and in Cape Cod as an approach to mitigate surface water nutrient pollution. So this figure here on the right is just showing like a schematic of a PRB placed in the subsurface where it can intercept groundwater and treat it before it enters a coastal water body. And a permeable reactive barrier, or a PRB, is an innovative solution that can be used to reduce nitrogen loading to water bodies. But PRBs are like one tool in a toolbox. And the unique thing about them is that they can treat nitrogen from all three of the main sources, including fertilizers, septic, and atmospheric contributions. The key is that it has to be in a form called nitrate. And after installation, this PRB treatment has no above ground structures. It uses natural materials such as sand, wood chips, and possibly other amendments such as biochar to promote the transformation of nitrate, which is our nutrient and in excess, it's a pollutant. And it converts nitrate to a harmless end product, which is nitrogen gas, which already makes up 80% of our atmosphere. And these barriers are called permeable because they're permeable to water, but they're a barrier for the contaminant. So the permeability is really a measure of how easily fluid passes through a material. And the PRB material is typically very coarse sand, wood chips, and gravel, and possibly future research will likely include biochar amendments. We're excited about that research initiative. And the PRB is more permeable than the surrounding soil, so that water is actually attracted to the barrier and then flows through passively and requires no energy input. And you can kind of think of a PRB like water flowing through a sponge. So it attracts the water stays in the barrier for a certain amount of time, allowing these reactions to occur. The microbes that exist naturally in the soil are just doing the reactions and using carbon, which is why we add wood chips. So again, the water is flowing naturally and then discharges into the pond without the nitrate. So to wrap it up and to summarize some of our preliminary findings, pore water conductivity and nitrate, as well as groundwater discharge rates and sediment characteristics, really highlighted that the northern half and western shoreline of Sagg Pond are the general areas contributing the most groundwater nitrate to the pond. And the discharge rates were strongly positive, but pretty variable in certain areas, and future work will involve our calculations estimating the shoreline discharge area and calculating nitrogen load at a finer scale using the results that using the data that we've collected this summer. And some of the main takeaways that we hope you get from this presentation is that land use and chemical makeup of groundwater is critically important for our local surface water quality, and really everyone has a role to play in either helping maintain or improve water quality in some ways. We see PRBs as a tool in the toolbox to tackle excessive nutrients. They're not going to work everywhere, and they require specific site characterization to determine their viability, but they are very promising for this area because of the high nitrate concentrations and the high discharge rates. And then, of course, other nitrogen remediation techniques are important and could include living shorelines and bioextraction. And of course, other necessary measures for reducing excess nutrients also include updating septic systems improved fertilization practices, especially for this subwatershed, which is mostly residential and agricultural. And with that, we'd like to thank you all for your attention. Thank you to the Peconic Land Trust and to all of our groundwater team members who helped collect all this data. Thank you to the Sagg community members and donors who have helped make this project possible. And thank you to everyone who's been involved.
And thank you so much, Molly and Ron. That was terrific. And I want to apologize to everyone. That has never happened to me before, but the chat function was disabled. So if anybody has anything they want to throw in, please do. I will, however, ask Melanie's question that she put in there, which is, does the opening of the ocean inlet create a suction effect that increases the groundwater flow and actually pulls in more nitrates at the north end of Sagg Pond? If Molly or Ron could address that.
Well,
Yeah.
go ahead, Molly.
Oh, yeah, so we did see those groundwater flow rates increase. And what we also saw when we were doing our measurements was that there was a flush of high saline water, like from the ocean, that did come in and really changed the chemistry of the pond, specifically in August, when it kind of coincided with a hurricane and a storm coming through. So yes, there is like high nitrate water coming in, and the ocean inlet opening kind of helps facilitate that high nitrate water coming in, but then also there is flushing. So it is a very complicated and dynamic system.
Great, thank you. If anyone else has any questions, please put them in the chat now because it is working. And if not, we will turn it over to Chris Gobler of Stony Brook University. And again, just throw any questions in there and we can ask Chris at the end or even get Molly and Ron back on. So yeah, I'll turn it over to you, Chris. Oh wait, Katya had a question, hold on. So Katya's asking, will installing septic nitrogen removal systems in the houses and properties surrounding the pond make a big impact on reducing the pond's high nitrogen levels? What are the current government programs and incentives?
I'm going to cover that, so maybe I can pick that up.
Okay.
John, we're a little over schedule. Do you want me to just proceed as I had planned?
Yeah, if you're going to cover that, let's do it.
Okay.
Thank you.
All right. Moment. Okay, good evening, everyone, and thank you for your attention. It's good to see many people I've seen before in prior presentations. And so going to give you some updates on what's been happening with the pond for this current year and some thoughts about things to do moving forward. Before I begin, I'll acknowledge the work of my lab group. We've probably had, I think, at least a dozen people out to the pond just this year alone, and certainly many more than that over the years. And if you're interested in what you're doing, you can, of course, follow what we're up to on social media. And in good news, Molly and Ron did a good job of covering a lot of the things I'm going to cover. So I can just go real quick through those. So the idea of the watershed, I think that was covered pretty well. And certainly the concept of what's happening across the land around Sagg Pond is influencing what's going on there. And I'll just certainly also mention, as was emphasized also in the last presentation, that as land use changes, so does the amount of nitrogen in groundwater. And that has consequences. And as an example, this is a map that I'm actually preparing the new one for 2021, but this is just showing 4 months of 2020 and different places across Long Island experiencing either state or federally defined water quality impairments, and that are driven by high levels of nitrogen. So for the past several years, I've been engaged in a research project, and management project for Sagg Pond. I think many of you have heard at least parts of that. And that's looking at how the water quality within the pond is changing over time, doing experimental work to understand what's leading to the overgrowth of algae within the pond, doing modeling studies to understanding the sources of nitrogen and phosphorus to Sagg Pond. We've done work on using DNA approaches to identifying pathogenic bacteria to the pond, but I don't have time to cover tonight. And also looking at filter feeding bivalves within the pond, specifically oysters. And so a lot of our observations have been made possible by a continuous monitoring buoy that we've installed in Sagg Pond. This is, I believe, our 3rd year of that installation. And for anyone interested, it's up on my lab's website. So at any point in time, you can go and look and see exactly what's happening on the pond and in the pond with time, and make your own graphs. And so I'm just going to cover just some of the things that are in there. Molly covered some of it with regards to something like salinity, for example. So ocean salinity is a number of around 32. So this scale only goes from 0 to 20. And of course, freshwater is 0. And so this shows June through, I guess, this afternoon data. And it does capture that second cut opening that actually was closed by, I believe, Tropical Storm Henri. But what you see is that that was not really a very effective opening. When Sagg Pond is open to the ocean and left open for a while and becomes tidal as it, over an extended period of time, you do get that salinity at this buoy that can get up to 30. And what you see here is a salinity that was very, very low, less than 5. It got to a peak of around 15, but then when closed, you can see within a week or so after that, got down below 10. And any number below 15 is a danger zone for something I'm going to talk about momentarily, blue-green algae. In addition to salinity, another thing we look at carefully within the pond is levels of dissolved oxygen. All living things need dissolved oxygen to exist, except for a few bacteria. So high levels of oxygen are important. And this is the oxygen data from this summer. You can see it's very dynamic. It may just look like squiggly lines, but this is actually quite natural for any coastal ecosystem. In fact, the bigger the range per day, the more intense the what we'd call the productivity in the pond is. And what you see over summer is actually a little bit of a troubling sign, in that oxygen levels were high and within the exact zone we would want in the beginning of summer. But you can see as summer waned on, the levels have gotten lower and lower. And since it's closed, since Tropical Storm Henri, there's been some very troubling signs. In fact, even in the last week, we've had extended periods of what we call anoxia, so almost no oxygen within the pond. And I've alerted Trustee Ann Welker. We were talking about this today, and I can say they're looking to reopen the pond within the next week, partly in response to this, but also partly in a response to another impairment that we look very carefully at and take very seriously, and that's freshwater cyanobacteria, also known as blue-green algae. I have a couple of micrographs here. The name Microcystis and Anabena will come up again. We worry about these organisms because they make toxins that are biotoxins and a human health hazard. For example, Microcystis is known to make microcystin, and Anabena is known to make anatoxin A. These are gastrointestinal and neurotoxins, respectively. When they were first discovered, they were called fast death factor and very fast death factor because they are very serious compounds. They've been implicated in things like promoting cancers. We know that they're harmful also beyond humans. I mean, that's in cases where you might be drinking the water. Thankfully, no one's drinking the water in Sagg Pond. But we also know this is a threat to pets. The CDC has indicated there's been hundreds of dog poisonings through the decades due to dogs consuming water with blue-green algae. And last year, there was even a case where elephants across the plains of North Africa were dying. No one knew why until they realized that they're actually being intoxicated by blue-green algal toxins. So these can be very serious compounds. And so through the summer, what we've seen, our buoy can monitor levels of blue-green algae by measuring the pigment called phycocyanin. So what you see here is that a pattern that sort of mimics the oxygen. This is what we've seen before in other water bodies, for example, Georgia Pond. As the blue-green algae blooms get intense, they have that effect on oxygen you already saw. So the buoy we use here is just measuring fluorescence via a particular probe, but we also have a device. We are the laboratory that's been designated by the New York State Department of Environmental Conservation for monitoring all of downstate New York for toxic blue-green algae blooms. And so we have a specific device that's calibrated with other such devices across the state. So with this figure, I can show you not only the levels of the blue-green algae, but how they measure up against thresholds already set by the DEC. And so what you can see is that when we got to late summer, we exceeded that threshold. When that pond was open, we had a little dip in the levels, but then the levels rebounded. This summer, we've detected and quantified 4 different potentially toxic strains of blue-green algae. You can see the 2 genera I already called out, plus 2 other ones, all of which have the potential to make toxins. This adds to the list of other species we've seen in prior years. I can say we hit a new record high level of microcystin in Sagg Pond in August at 1.2 micrograms per liter. So in bad news, this is above the drinking water standard set by EPA, but it's below the recreational standard. But still, I should say, once the DEC sees a level above their standard, their goal is to close a water body for recreation. So obviously this is a situation we do not want. And so the question is, why are we having these blooms? And to address that, we've conducted some experiments looking at the relative importance of nitrogen and phosphorus. These are bioassay experiments following fairly standard protocols, slightly modified for some of our local systems here, but essentially we're setting up experiments looking at nitrogen and phosphorus in both compounds. And so when we conduct that experiment, essentially what we see is that when we add nitrogen, we get a significant increase in the amount of blue-green algae in the pond. Phosphorus has a very small effect, although both compounds together have a modest increase over the control, which is where we're adding nothing. But from this, we basically conclude that nitrogen is the most important element for controlling algal blooms within the pond. So from there, we want to understand where is the nitrogen coming from. And so last year, we finished our models looking at the different sources of nitrogen. In addition to doing just generally modeling studies, we did a full sediment study to understand the flux rates and quantify the flux rates of different nitrogen species out of the sediments. So that involved a full sediment survey that was done. Actually, I think the surveys were done 2 summers ago, which match up very well with what Ron and Molly had shown with regards to the depth of mud and the relative organic matter of the sediments as well. But the important thing is that the study found, and I'm sort of glazing over some of the details, but that the largest sources of nitrogen were both agriculture and then on-site septic systems. And we're still looking to refine. We think that agriculture number might even be a little bit smaller because of updates, some farm fields being fallow and some farms that were active being less. We also mapped out groundwater measurements made by Suffolk County, which actually also aligned very well with what Molly and Ron found, and that is very low levels of nitrogen in the lower ends of the ponds, but higher levels up in the neck on both the east side. Just to briefly talk about the septic systems, I know Katja had asked about this, and so, and again, this represents about 1/4 of the nitrogen entering the pond. And so for those of you who are not familiar, the current array of septic systems for each home on Sagg Pond is not far off from what we see here. In fact, this is probably the most advanced type of system that might be there. In some cases, an older house might not even have a septic tank. But the upshot is these systems are designed to allow all the liquid waste from homes pass right into the groundwater. And as Ron and Molly emphasized, that then is translated and transported directly to the pond. So Molly had showed the groundwater travel time, but this is, I want to emphasize Suffolk County's wastewater sub-watershed study that was released last year. And the two things I want to really emphasize here is that when it comes to wastewater in Sagg Pond, Sagg Pond was put at the head of the list for places that should be upgraded. You can see here they had a ranking of 1 through 4. So Sagg Pond was defined as sensitivity rank 1. So most important for being upgraded. And you'll see down here in the lower left, they recommended a 63% decrease in nitrogen load for this system. So in good news, not only did Suffolk County complete this study, but they also have approved a series of what are known as innovative and alternative septic systems that can be put in to homes to reduce nitrogen levels. I'll also mention that another hat I wear at the university is I'm director of the New York State Center for Clean Water Technology, where we're working to harness science to engineer clean water for the protection of public health and the environment in New York and beyond. We have a goal here we call 10-20-30, where we're seeking to create systems, septic systems that get nitrogen down as low as possible, ideally below 10 milligrams per liter, and will last for at least 30 years and will cost less than $20,000, which is the amount of money available for grants currently, just in Suffolk County and New York State alone. And so we've created something we call nitrogen removing biofilters. In fact, I'm proud to say that Molly worked on these during her PhD at Stony Brook when she was working within the center. And these are very simple systems that are meant to be attached to existing septic systems. Some people also call them the layer cake system because they're very, very simple. They're made with natural products. They're not commercial products. It's just setting up what's known as a drain field and underlaying that with a layer of sand and then a layer with sand mixed with wood chips. And that allows the microbes that are in the ground to actually remove the nitrogen in a way very similar to what Molly had described for her permeoreactive barrier. We've published on these systems in international peer-reviewed studies. In fact, let's see, Molly is actually a co-author on this paper. And we're very proud of the fact that these systems are performing as good or better than any system tested in Suffolk County. So what you're seeing here are the different types of systems that have been tested in Suffolk County, and the average of those systems is in the black bar all the way to the right. There's many systems that have been tested that don't even meet the Suffolk County standard, which is about 19 milligrams per liter. I think my line might be a little off there, but the bottom line is our systems are as good or better than almost any system out there. And that some of the systems that are out there are sort of just on the cusp, kind of making the grade, not exactly necessarily making the grade. So one thing that we're eager to do as a center is to help Sagg Pond and think about the idea of having for Sagg Pond a watershed septic manager, to someone who could organize, expedite, and oversee the mass installation of innovative and alternative septic systems, ones that are going to be maximally protective of the environment and would address a quarter of the total nitrogen loads into the system. And right now, this is very much a commercial enterprise. So what the center would bring would be the best science possible to improve upon standards. So optimize septic systems for each individual property, having the right system for the right location, coupling those systems with other systems that we've made called nitrogen polishing units, and also even installing nitrogen sensors that can monitor and assure that the systems installed are performing correctly. And then also to assist with the very extended and circuitous design, permitting, and installation process that goes along with this. The last thing I'm going to talk about is the work we've done with oysters in Sagg Pond. For those of you who do not know, Sagg Pond is neighbors on Mecox Bay. And there's actually some very interesting parallels between Mecox Bay and Sagg Pond. They represent 2 of the 3 systems that are temporarily open to the ocean, along with Georgia Pond. And Mecox Bay currently has Long Island's most abundant oyster fishery, and it has had that for decades. And the reason is that the temporarily open / temporarily closed nature of these bays actually make them ideal habitats for oysters. Oysters don't like full ocean salinity. They sort of like that brackish salinity that you can't find in most Long Island estuaries. So the low salinity that you find in Mecox and Sagg Pond is pretty unique. You don't see that elsewhere. And so that's one of the reasons we think that Sagg Pond could also be a great place for oysters. We're interested in things like oysters because they're what are known as ecosystem engineers. They can filter water, keep water clean, and actually help mitigate algal blooms. And in fact, the figure below is from an experiment I conducted many years ago where we had a tank of water. That brown water is a harmful algal bloom. Both of these tanks were exactly the same a week prior. This one had a dozen oysters put in it. You can see what the outcome was. These are 300-liter vessels. So we've been putting out oysters in cages over the last 2 years and essentially monitoring their growth, their survival, and their disease. In 2020, we had 2 different sites, one towards the northern end of the pond and one more towards the south. And we had a lot of trouble with that northern site. It was right by the bridge there, and I guess people were always very, very curious as to what was in there. The oysters were not edible, but we had a lot of trouble with those cages. So we did not get far with those experiments, but we did quite well with the one in the bottom. So in 2020, we had 2 different cohorts of oysters that we had put out within Sagg Pond. One in July and then one in August. Whereas, and those 2 were one-year-old oysters and then ones that had just been spawned. Whereas this year we put out, again, 2 different cohorts, but tried a new approach. So, spat on shells. These are very small oysters. This is a singular Atlantic surf clam shell. And on it, you can see there's several dozen oysters. And that might seem weird, but as it turns out, in the wild, oysters don't grow as individuals. They're actually colonial organisms that grow as something known as oyster reefs. And so in the end, that's the kind of the manner in which we think they would grow best in a place like Sagg Ponds. We tried that this year. Now, last year, just to show some quick data, the first deployment of the oysters, almost none of them lived. It happened to be during a blue-green algae bloom. We then did our second deployment, and in doing so, we saw much better survival at the southern end of the pond than the northern end of the pond. And then for this year, we've had significantly better survival of the spat-on shell than the seed oysters. And that is despite the fact that there's been anoxia and these blue-green algae blooms. So there may be something too, and we have a lot more work to do to analyze all this, looking at growth and other sorts of things. But we think there's probably something to and value in trying this spat-on shell approach. So I'll just wrap up then. What I showed you is that Sagg Pond continues to experience these blue-green algae blooms by multiple genera of blue-green algae, and that they're producing microcystin. And so that's probably the #1 environmental concern in the pond because that's a human health hazard. I showed you also that this year, in fact, just in the past 24 hours, the oxygen levels are getting very low in Sagg Pond at night. And so hopefully that pond, the pond will be opened to the ocean by the trustees in short order. I showed you that the algal blooms that we worry about are promoted by nitrogen, and that the biggest sources of nitrogen to the pond are on-site systems and also agricultural fertilizer. And we've also discovered that oysters can survive and grow in Sagg Pond. And we're now trying to optimize strategies to ensure figure out the precise age and developmental strategy for putting them out. And then for next steps, I mentioned the Sagg Pond Watershed Manager. I think that's something that can be really valuable in moving the whole community towards having a cleaner ecosystem. And we're going to continue to look at the mass culture of bivalves along with bioextraction. We do a lot of work in my laboratory on harvesting different macrophytes for different types of ecosystems, and we think there's an opportunity there as well. And we'll obviously want to do whatever we can to collaborate with Ron and Molly as they move forward and looking at groundwater. And I'm glad Debbie Aller's here because there's probably an important role for thinking about best practices for current fertilizer use, both for homes, but also for agriculture. And so with that, I'll thank you kindly for your attention.
Thank you so much, Chris. That was wonderful. I don't see any questions in the chat. If anybody wants to throw anything in, and as I just wrote in the chat, like we're happy to answer questions after the event, just either respond to the event invitation or reach out to one of the staff members if you have our email, whatever way you can get us. So yeah, that being said, John, I'll hand it back to you for wrapping it up.
Well, I want to say, first of all, thank you to all of you who have written checks, made donations to the trust to underwrite this work. A tremendous effort on the part of all of you that started out supporting Chris's work and most recently helping to underwrite the costs of the Cornell work. I just want to say that the data that all of these projects are generating is really intended ultimately to inform a management, a revitalization plan for the pond. And that is to be ultimately prepared by the town trustees. They have received some town money for that purpose. And I don't think that Anne is here this evening to really speak to that, but we'll be talking with her in the coming days and weeks to just get a status report on where things stand with that. But of course, as you can see, the data is still being developed to inform that plan. So it is definitely a work in progress. But as a consequence of what we have heard this evening, it's clear that the data really shows and reinforces that nitrogen is the key problem. And the sources of that are fertilizers, both agricultural and residential, as well as the septic system. So that, next steps really need to be, first and foremost, to complete the work that Cornell is doing in terms of site characterization that will really inform us as where the best locations are to mitigate the flow of nitrogen coming in through the groundwater. That's one piece of the puzzle. And also the concept of a septic manager for the Sagg Pond watershed, I think is a great one in terms of helping landowners, property owners, find the right system that really will be the most efficient. I think one of the things that Chris pointed out is there are lots of systems, and some are less efficient than others, and a lot of that has to do with the specifics of the property on which these are going to be installed. And I think that the concept of a septic manager to work with property owners in the watershed is a very good one, and one that we certainly want to support. I think the key to our success up to this point has been the infusion of private dollars from the community as a whole. And that helps us leverage public money. And we do have, as Ron mentioned, a proposal before the CPF water quality program, and that will be up for a public hearing on October 16th. We have funded, with your support, about $50,000 of an over $200,000 project cost for the phase 1 and 2 that Ron and Molly are doing. The idea here is that we've requested 50% of the overall cost from the town through the CPF, which means we would need to raise, assuming we get it, the full amount that we've requested, another $50,000 to complete the Cornell work that is currently underway. So that's going to be one of our goals before the end of the year. I want to talk more with Chris about the funding of the septic manager position. That may be another component of what we're going to add to a goal for year end from a fundraising standpoint. And then, of course, your support of the trust to coordinate and move all of this forward is very important as well. So I'm going to just give us a general goal of 150,000 to raise by year end that will finish the work of this phase of Cornell, assuming again we get what we need from the town through the CPF. But I think we have a very compelling argument because of the record of private funding that we have brought to the table. And then again, the septic manager to move forward in an efficient and effective way on the installation of septic systems, I think is another great piece of the puzzle. So more on that in the coming weeks from us. We will sort of formalize what our goal is for the rest of this year in terms of fundraising and the specific outcomes that we expect to get from that. And I can't thank you all enough. I shout out to Katja and the Goldman Family Foundation, because they've actually provided a lot of the seed money for this, and you have matched it. So it's very helpful to have people who care and who will put up resources to really encourage additional people to support this work. We now have, I think, a database of well over 300 people within the Watershed email addresses. We're building that every opportunity we get. And, to mobilize you, we're going to reach out to you as we get closer to the October 16th public hearing to really encourage you to reach out to the town board member. We'll give you a sort of a general draft letter that you can adapt, but to send that to the town board because it's their final decision as to what gets funded through the Community Preservation Fund. So your support and help in that regard will be much appreciated. But you'll hear from us in the next few weeks as to who and how and what to say in terms of supporting this effort. So on that note, we've sort of gone a bit over time, but I think it was well worth it for you to have an opportunity to hear what you have underwritten thus far in terms of moving us forward to a cleaner, healthier Sagg Pond. Obviously, there's a lot of work to be done. It's not going to happen overnight. There are multiplicity of factors that affect the health of any water body. Chris, Ron, and Molly can attest to that. It's a matter of really understanding the dynamics of each coastal pond. They're all different. And what we're getting with their work is a picture of how Sagg Pond and its watershed function. So I really appreciate you coming this evening, and we'll be in touch again soon. And please do send us any questions. I see, what do we have here? Anything there that we should bring up, Amanda?
No, Chris really addressed them, and I took note of all of them, just if there are other answers to them that we need to address later.
So we will respond to each and every one of these instead of taking everyone's time right at this moment. But we will get back to you and share a response to any questions that are outstanding. I do see one though that might be worth mentioning because it wasn't really touched upon, and that's the question about the role of birds. And that sort of goes, I think, to your work, Chris, that you did previously. You might want to just give a quick response to that one.
Yeah. So the bird on the nitrogen front, as it turns out, the birds are really not of consequence. However, when we did our microbial source tracking work and tracked the DNA of pathogenic bacteria within the pond, they actually were an important source for those pathogenic bacteria. And that seeming discrepancy is actually not surprising, in that what that is telling us is that the bacteria are likely being delivered via surface runoff, whereas the nitrogen is being delivered within via the groundwater, as Molly and Ron emphasized. And microbes are generally retained as they flow through groundwater. So, so birds important for adding bacteria to the pond, but not really for nitrogen, which is seemingly a paradox, but is actually has come up in many other systems on the East End as well.
So thank you, Chris. And thank you, Jackie Fenlon, for correcting me. The public hearing is October 12th at 1 p.m. And if I'm not mistaken, I think it's an in-person one if people do want to go, although who knows, given this variant, what the world will look like in mid-October. But we will give you more information about that following this meeting. So October 12th, not the 16th, at 1 p.m. at Town Hall. Via video conferencing. Okay, good. I see that. That's great. All right. Thank you again, everyone, for being here. It's great work and complicated work, but we'll get there. And all of your support, again, is much appreciated, and you'll be hearing from us again shortly.
Yeah, thank you, everyone. Have a great night.
Thank you.
Good
Thank
night.
you.